Okay. I guess the room is getting full, so we're gonna get started. So for those of you who don't know me, I'm Javier Gonzalez-Castillo. I'm staff scientist within the section of functional imaging methods. And today we're gonna be talking about connectivity and dynamic connectivity. And the talk should have been called functional, because that's all I'm gonna talk about. There is then the whole world of structural connectivity, which is also very important, and you can definitely explore with MRI. There's not going to be anything about that here. So I thought that for an hour, we should just, talk about a few things because there is not that much that can be explored in an hour. And those are the learning objectives for today. I'm going to try to define what functional connectivity is and also an emphasis on what it is not. Then we're going to go into different ways in which you can estimate functional connectivity. Then I'll try to get into the differences between dynamic and static functional connectivity and explore a little bit the ways in which people look at dynamic functional connectivity. And even though a lot of people think of functional connectivity as being just about resting state data, the truth is that you can also do it with task data, but you have to have a couple of extra thoughts or special considerations when doing that. And that's what's going to be the last part of the talk. Of course, please stop me at any moment if you have questions, or you can also ask me at the end. So what is functional connectivity? So perhaps the first paper that mentioned this concept of functional connectivity within the framework of fMRI is this one from 1994 by Carl Friston. And he tries to put it within this context of thinking about the functional organization of the brain and having these two principles of functional segregation and functional integration. Functional segregation being the idea that you have different parts of the brains that are specialized into doing something. And that's usually what we go after when we do task-based studies. And functional integration, the idea that for a behavior or to achieve a goal, it's not that these areas are going to be working separately, but they need to talk to each other. And that's what we're going to try to look into when we look at functional connectivity. Within functional connectivity, in that seminal paper, he makes a very important differentiation between what is functional connectivity, which he defines as the temporal correlation between remote neurophysiological events, basically in fMRI jargon, between different ROIs or boxes that are far apart. And effective connectivity, which is a much more precise term, that this idea of the influence that one neuronal system exerts over another. This is kind of hard to measure with fMRI. Some people would argue it's possible, but there are a lot of problems that derive from the idea that in fMRI, we're not measuring directly neuronal activity, but we have that hemodynamic filter in between. So again, this is something that I'm not going to talk about here. I'm going to focus just on this basic, although very widely used concept of functional connectivity. And if there is only one thing that you should take home today, is this idea that functional connectivity is simply a statement about correlation. Okay, we're going to take time series and we're going to look at the temporal correlation between them. Maybe we'll do something fancy like ICA. Maybe instead of correlation, we'll do mutual information. But it's just a statistic. And what's causing that observation can be many different things. It can be just a transient event due to an stimulus. It can be truly that these regions are talking to each other, which is what we would like to infer. But it can also be that they just have a common input that's causing them to have a common signal, but it's not like them talking to each other. So as you prepare your discussion, when you do your first experiment with functional MRI, always keep this in mind so you don't overinterpret the results. I think that's something very important. Okay, so how do we measure? Well, the definition at the time had the word correlation, so that's how we tend to measure it. So if we have fMRI data across the brain, we might pick the simplest way to do it. We might pick a seed, let's say in visual cortex. We extract the representative time series. Of course, this is done after you denoise your data in the best possible way, which I believe there's been some talks about that. And I'll talk a little bit later about some extra emphasis that you should do when you're thinking on doing functional connectivity as opposed to task-based analysis. And then once you have that time series from that seed, you can compute the temporal correlation, Pearson correlation between that seed and every other voxel in the brain. And you get a map like this where the color of a given location tells you how strongly the signal in that other place correlates with the one in your seed. So it looks like here in frontal cortex, we have a region that correlates very strongly with our seed here back on occipital cortex. It looks like here in motor cortex and frontal temporal regions, there's not that much. Now, this map that we see down here will of course change the moment I move the seed somewhere else and somewhere else. So seed-based, I really like it because it's a very straightforward way of looking at connectivity, but you really need to have an hypothesis. Otherwise, it's really hard to go all over the brain and really see what's going on. Now, that said, it's always a good idea to explore, not just because of, you might get to new hypotheses, but also because a seed-based analysis is also a very good way to actually detect if our data is clean or not. And for those of you who are familiar with AFNI, there is this functionality called Instacore that I think comes really, really handy. And it's also a really good way to learn neuroanatomy, because when you see blobs, then you go and find where they are, and it's fun. So what this does is you can basically open AFNI, you select a data set. You can even do it with data sets as they come out of the scanner, because in this way, you're going to have the opportunity to do some very basic denoising, like a little bit of spatial smoothing, despiking, bandpass filtering. You can even put some regressors to orthogonalize the signals against them before you compute connectivity. And then you press go. And in a couple of seconds, or a little bit more if you have really big data sets, you'll get something like this, in which you can put the mouse somewhere in the brain, and as you move around, as you will see in a second, it recomputes the connectivity between where the mouse is and everywhere else in the brain. So here we're starting with me having set the seed right here in occipital cortex, and seeing that, since there is a lot of correlation between occipital cortex, but you will see in a second, as the mouse starts moving around, how the pattern of connectivity changes. So now we have more lateral occipital. Here we're able to find the auditory network. In a second, we might go and see the motor network. So this is a really clean data set where you can see that as I move around, the networks that we're used to see appear very, very easily, but that might not always be the case. Still, even here, I'm just doing it really fast, but I'm really moving around the brain. Every time I get to a new location, I get a new map. So it's hard to really explore all at the same time. So another way to do this is to rely on a brain parcellation. Again, we have our data, but now we take a parcellation, a pre-existing one, the Suffer, the Glasser, you name it. There is so many out there. And we extract the representative time series for each of those ROIs, and then we can construct a functional connectivity matrix. Basically, what you have here, it's ROIs by ROIs, and the color of a tiny dot here represents the Pearson correlation between the time series of these two ROIs. If you get to sort this matrix in a way that makes sense here by networks, you can start to see how regions that are within the visual network, they're very much connected to each other, the motor network, the different attentional networks, and the default mode network. Once you have a matrix like this, you can set a threshold. You say, I'm only interested in correlations above 0.5, whatever it is. And then you get an adjacency matrix that basically has zeros and one. And from there, you can go into the world of Josh Paskovich, which is your next speaker. Think of it as a graph and start doing anything that you can get out of network neuroscience or graph theory. And there are many different tools that you can apply. But at the end of the day, it comes just from having first computed those temporal correlations between the time series of all the ROIs, come with the connectivity matrices, threshold it, and then move into the graph theory world. And that makes sense? Now here, we have these predefined ROIs that come from atlases that hopefully they've been done in a very precise way, but they might not align very well with the functional edges of a particular subject. So we might be interested in saying, hey, I really want to explore all the data, but I want to do it on a data-driven way. One way to do that, and the most common way, is to rely on independent component analysis to study connectivity. In this case, what we're going to do is we're going to take our data and we're going to abstract it as a 2D matrix, where we have a space, meaning voxels in one direction and time on the other one. And by using ICA, we're going to decompose that matrix into signals that are statistically independent. And for each of those signals, so then basically we come up with two matrices. One has components by time, and the other one has components by space. So for each component, which gets selected automatically, or you can say 130 component, and that's a whole world design how to do that, you're gonna get a representative time series and a spatial map. And here is an example of one of the many components in a given data set. Here it looks like this is the default mode network, which we know because of its anatomical presentation here, a representative time series, and then once you get the representative time series, you can always compute the spectrum, which is a good idea. So you have a resting state data set, you do ICA, you ask for 100 components. So now you have 100 spatial maps and 100 temporal time series. You could think, okay, now I have all these signals that I know they're present in my data. I'm just going to compute the Pearson correlation between all of them and come to have a connectivity matrix in which, instead of depending on a functional parcellation, I've come to this functional parcellation being driven by the data. So there are two things to keep in mind, and nothing is wrong. One is when you take an atlas, the ROIs from which you extract the representative time series tend to be contiguous voxels in Spain. Here, you're going to have a single representative time series for all these voxels that are not necessarily contiguous. So you're going to be looking more at connectivity at the level of network to network, not ROI to ROI. Again, it really depends on your hypothesis, what you want to study. And the other thing to keep in mind is that when you do something like this, no matter how good you've been at preprocessing your data, there's going to still be some components that are noise, that are not really most likely of neuronal origin. And the first thing you're going to have to do is to really try to get rid of those first. So let me give you some examples. So here we have, again, same data set most likely. This is another component that looks like the default mode network. We can see it because, it sits primarily in gray matter regions and follows the regions that we know are part of it. It has a time series that seems reasonable, and the spectrum of that time series seems to be the power below 0.1, which is what we expect for resting state networks. But here is another component that looks like most of the signal comes from the edges of the brain, and definitely the profile in terms of the spectrum is very different. We have these very high components here that are kind of weird. And then we see this big dip here. Well, something like this most likely comes from motion. And if we actually compare the representative time series of this component to the motion estimates, we can see that that dip aligns. Here is another example. We have a component that clearly it's sitting not on gray matter, but mostly it looks around the ventricles, and it has this kind of profile that is very, almost like a sinusoid, very repetitive. We look at the spectrum. So this is something that's usually associated with a result of cardiac and respiratory function, some form of physiological noise. Here we have one that's about susceptibility artifacts. It tends to appear in these frontal regions. And again, the time series are very weird because it's not like the one I was showing you that is kind of a smooth of the default mode network, but you have these weird dips. Not to mention also the power is just concentrated really, really low. So we're going to have to get rid of those first before we then go ahead and compute connectivity, because otherwise we're going to have a matrix that contains these correlations between things that are not really informative. Now, I described this in less than 5 minutes, but you say, how do I go if I'm new into really deciding what's a good component or a bad component? Because it's not that easy. Some of them are really easy to see, but some of them are not. Well, one way is to really look at a lot of them. And over time, and talking to other people, you develop an intuition of what it is. And this is a good example of how to go through kind of a process of looking at the spatial map, at the temporal, at the spectrum, and there are some characteristics like sitting around the edges, sitting on the ventricles, not being really smooth. There are several things that you can do to decide. But over time, people have also developed ways to do that automatically. So there are two main ways to do it when you're dealing with single echo fMRI. One is called ICA aroma, that is mostly geared towards finding components that are related to motion artifacts. And then this other one called FIX that tends to look at all type of artifacts. Those inside, basically what you have is a classifier that has been trained, looks at specific features of the components, some of them very similar to what you have here and some others. And then it throws out basically a listing that tells you this is a good component or a bad component. And then you just keep the good components and move forward. It is always a good idea to double check the output of those things. And I understand if you have 1,000 subjects, you're not going to do it. Just pick randomly to see if there is something weird consistently. If you just have 10 or 12, it's a good idea to look at all of them. Now, if you're lucky enough to be working with multi-echo data, which I think there was a talk about it recently, Then there is this other approach called TEDANA that tries to do this automatic classification of components, taking into account that signals that are of both origin, therefore more likely to be neuronal, or not all of them, have a specific profile across echoes, that signals that are not bold, like scanner artifacts, head motion, and use that additional information to try to separate good from bad. That makes sense? And then once you pick your good components, we can go back to constructing a connectivity matrix like I was doing based on the parcellation. As I said, the main difference here is that each of these cells now is not the temporal correlation between two contiguous sets of voxels. It might be between all these and all the green areas here. So you're looking at connectivity at a more high level view. All make sense? Now, the way you compute this connectivity matrix is by taking the representative time series of all these components and computing the Pearson correlation. So we go back to the basics. And we all do Pearson correlation because that's how we all studied, but maybe there are other ways to do this better. And there are some papers that have looked into that. This is one particular one. They look at comparing full correlation with different types of bandpass filtering to different ways of computing partial correlation, which is kind of the similar idea, but instead of just looking at the temporal correlation between two ROIs, is the temporal correlation between these two after taking into account everybody else. So it's a little bit more computationally expensive. You can think about using something like mutual information, which will look for something that are not necessarily just simple linear relationships. There's been a lot of work looking at things that actually look at that concept of effective connectivity, a relationship between the signal and a lag version of the other one, coherence, and a lot of other ones that I don't even know what they are, those in particular based on Bayesian nets methods. And in this paper, they came with a way based on simulations on putting a number on how good a particular metric is at detecting connections that are truly in the data, because those are simulations they know, versus the ones that shouldn't be there. And what we can see here is that full correlation performs really well. There are a couple of things that seems to perform a little bit better, basically partial correlation and these Bayesian-based methods, but overall, just simple correlation, which is really fast and easy to compute, does a really good job. And that's, I think, one of the reasons that we keep relying on it a lot. In this study, it's not about just how well you're able to detect the true connections versus the bad connection, but also how stable that is compared to the different things that you need to take into account or make decisions when you're collecting data for doing a connectivity analysis. And one of the things that seems to matter a lot from this point of view of looking at connectivity in the ways we've been discussing so far is the duration of the scan. And that's a question that comes very often. How long should my resting state scans be? If you ask me, I'll say the longest you can. But I know that's not a valid answer because we all know we have just 2 hours there and we need to put a lot of things. So in this study, they said that really makes a difference to go at least to 10 minutes. You start to find a lot of stability in your connectivity measures when you hit those 10 minutes instead of just doing 5 or 6. We look at that ourselves too in a different way. And here the take-home message is that whatever we're looking here, which is the reliability across different, we scan people for 60 minutes and we carry it in chunks. And then we look at the reliability of the connectivity based on all these chunks. And it looks like once you start hitting that duration of around 10 minutes is when you start to see more reliability in these different chunks of the same person and across people. Here is another study from another group, around 13 minutes. So it looks like, if you can do something at least 10 minutes, 12 minutes, if you're interested in connectivity. The other thing to really pay a lot of attention, especially for those of you that come potentially from task-based study when you move to functional connectivity, is this idea that because we're just looking at correlation between different spatial things, anything that can induce a spatial correlation that looks meaningful, it's a problem if it comes from something that is not neuronal. So any source of noise that we know can lead to that, we're going to have to be very careful about trying to remove it in advance. So for example, if we're taking data with multi-coil coils, and we have one of them that is kind of flicky, we might get data that contains spikes only in one part of the brain, and you might get a profile of connectivity that looks like this. Clearly, this is an example that you would throw away the data because that's not something that will happen. But there are ways to correct for that. There is these methods called anaticor and fast anaticor that use an additional regressor that we don't tend to use in task-based, which is basically the signal in a little set of white matter around each box. The idea being that white matter should not contain neuronally related signals, but any little flicker on the coil will affect the region. Therefore, by using a regressor that comes from the local white matter in every place, you get to clean up the data, and then you get a connectivity map that is more like you would expect. Head motion, it's a gigantic problem for resting state 2, especially because head motion seems to have this effect that affects differently long-distance connection and short-distance connections. So in general, the more head motion you have tends to lead to a decrease in long-distance connectivity. Here we have an example in 2 different subjects in which connectivity between the PCC and frontal regions of the default mode network is not present unless you do some serious scrubbing, which means throwing away volumes that have excessive motion. And usually that threshold that we mark to say those are bad volumes needs to be a little bit more conservative when we're dealing with connectivity analysis versus task-based. And it applies the other way around. For the more motion tend to increase local connectivity. And this is an example in which they saw that if you do a scrubbing based on motion, then you lead to a change in which you see these 2 effects, while if you do a scrubbing in a random way, you don't see that. There is the whole world of physiology. I mean, those subjects are breathing, their heart rate is beating, and that can lead to patterns of connectivity related to variation in respiration rate, variation in heart rate, and some systemic vascular waves that really look a lot like connectivity profiles. And that's why when you're gonna do some connectivity, it's really, really important if you can to acquire on top of your fMRI data, concurrent measures of physiology, the respiratory belt and the pulse oximeter at least. If you don't have those, there are ways to extract physiological noise by looking at the components in the ventricles. There are tools that you can use. But if you're studying a new study and you think you're gonna look for connectivity, I think it's a good question to see how much extra work would be to acquire those signals. And here at NIH, it's in all the scanners, no? Pinat, Siemens, and the. And then there are many other factors that affect connectivity. You know, our subjects tend to fall asleep, especially during resting state. Here, what this graph shows is that the proportion of subjects that are going to be awake tends to go down as your scan goes longer. And that's important because connectivity changes a lot between being awake and being drowsy or asleep. It changes in particular in some networks, but also there is an overall tendency to have higher connectivity when subjects are drowsy. Now, what can you do about that? Not that much, but you can try to at least have an expectancy of what has happened. You can either do concurrent EEG, and that's the overkill, or you can maybe just ask them or look for some of the features that we know are present on the data. Especially if you're going to be looking at studying what you're comparing to population, make sure there is not a systematic difference on these factors. We in our group are very interested now in starting to look at another factor, which is this idea that different people, when they enter the scanner for a resting state scan, they might have a different experience, they might have a different set of spontaneous thoughts, and that can have an influence. Again, do you need to worry about this on day one? No. Now, it's something to keep in mind, especially if you're going to be comparing two populations that you know one of them is very propense to having a specific type of spontaneous thought. Might be a good idea to have a questionnaire at the end. Is all this clear before I move to the next topic? So, everything I've talked so far, even though I haven't been using the word, it's static connectivity. Why? Because everything we've been talking so far, whether it's computing this connectivity matrix or doing ICA or doing whatever it is been always taking into account the whole duration of the resting state scan. But one could think, hey, why don't I chunk it? Oh, look at what happens. You know, you can, if you have particularly a long scan, you can say, oh, let's look at the connectivity during the first 60 seconds, another 60 seconds, another 60 seconds. And what you will see repetitively, and this has been shown in many different ways, is that connectivity change. Is that what you see in your static connectivity is not something that is continuously there. It's not like the regions of the default mode network, they're continuously talking to each other. It's overall, they tend to have strong moments in where they're talking to each other. That tends to happen often enough that on that average view, which is what you get out of static functional connectivity, appears very clearly. but sometimes they'll talk with one region, sometimes they'll talk to another region. Now, when you start exploring connectivity and how it evolves within the confine of a single scan, that's what we tend to call dynamic functional connectivity. And lately, because the word dynamic has some implications from a statistical point of view, it seems most people is converging into this concept of time-varying functional connectivity. So just to make this point clear, why I think it's interesting to explore dynamic functional connectivity. It is one scan, I believe it's a HCP or a long 15-minute scan in which we have cleaned up the data completely. And I have put a seed on the posterior cingulate cortex, and give or take, you get a beautiful DMN. You know, all these regions in the frontal and parietal are connected to each other. Now, what I want to show you here, it's the evolution of the time series that have contributed to this data. It's the same data, but here what we're going to look is how activity goes on and on over time. The red dot here is going to show you, where we are in time. And the signal that you're seeing is just the signal at this particular voxel, which is what I use as the seed to compute this static connectivity map. C-plane, yes. So what you can see, there are these big moments. What you can see going in red, these areas of the deformable network when just happened. But you can see it's not all the time. And that sometimes that goes with some other areas. So let me just show you examples. So here is one moment. Now what I did is I stopped the video. I'm just showing you some of them stopping, in which the deformable network seems to be on. Here is another one, slightly different, but the same. Another one. So it's true. I mean, the deformable network we've seen in a static connectivity, because it's true that often and strongly these areas are going together. But it's also true that here I go to another moment of peak activity in this region. It seems to go with occipital cortex. It seems to go with motor cortex. It seems to go with this other medial region. And I think that this is also informative. The question is, what's informative about? And it's this type of information that we're only going to be able to access if we open that window of dynamic connectivity. That makes sense? So how do we do that? Well, the first thing we can do, which is the most often used thing, is this idea of using a sliding window correlation. What we're going to do, if this is our 10-minute scan, we're going to pick a window, and we're going to compute the correlation of every single ROI or voxel or ICA component, just using the time series on that window. Then we're going to move the window a little bit, and do that game again. So as you can see here, now we have two more things that we have to decide as we're doing our analysis. We have to decide on a window length, and we have to decide on a window step. What to use? Still open out there. Usually, for fMRI data, quite around 2 seconds, I would say a window of 30 to 45 seconds seems to make sense. Seems to make sense, meaning it's one that's used often, that people have been able to find relationships to behavior, that you have enough data points that, your correlations are not going to be extremely noisy. But it's not like there is a perfect number, because think about it. If something of interest is really, really fast, and I have a window of 45 seconds, I'm just going to miss it. So it really comes to the temporal scale of whatever you're going after. But once you do that, for every single connection, instead of having a single number, you get to have a time series of how connectivity evolves, which when you think about all of them against each other, what you end up having is a set of connectivity matrices that tell you how the different snapshots of how the brain is connected as you move through time. These are two. So what I'm doing in some of the slides now moving forward is pointing to references that might help you, have a more informed view of the issues or how people do these methods, because there's so many ways to do dynamic functional connectivity analysis. It's not really possible to talk about it in 1 hour. So those, I think, are not the only, maybe not the best, but some of the references that I think are a good starting point for those of you who might want to use a sliding window and think about the issues that comes with it. But once we've done it and we're confident that we did it properly, once we have these matrices, one thing you can do, and people have seen that, is that if you look at them, it's not like all of them are different from each other. There seems to be these patterns that they reoccur over time. Some of them, they happen now, they happen again, and then later, and here is another pattern. So some people have spent a lot of work, including ourselves, in finding ways to extract those patterns. And once you have those patterns, And then you can do some temporal modeling, like thinking about which pattern happens more often. Is there a pattern that is always followed by another one? What's their dwell time? Many different things you can do. For those of you interested in that idea, these are good papers. This is kind of the original paper, and I will explain a little bit of the paper in a second, that did this business of taking a sliding window correlation and then applying some clustering to find those functional states. This is a follow-up that we did ourselves, trying to see whether they were related to something meaningful, using a multitask scan in which we induce some mental states by asking people to do different tasks, but scanning them continuously. And we were able to find that, yeah, different functional connectivity states seems to relate very well to the different tasks that people were doing. Now, the other way to think about exploring the dynamics of the signal during a resting state scan or potentially a task scan, is instead of looking at correlation, it's like going to the time series and finding the moments where you see big peaks of activity. Hopefully, because you've done your denoising, those peaks are truly neuronal. And then selecting those and looking, if you had a seat on the PCC, potentially then looking what's happening everywhere else in the brain at that particular moment in time that seems to have a strong signal. This has been done in different ways. There is this idea of point process. There is this idea of using the convolution, which we did some work that actually takes advantage of multi-echo data to do that a little bit more properly. This is just another way of not just selecting peaks based because they're big, but because they correspond to a moment where an hemodynamic response function would fit well the data. And once you find those peaks, you can, as I say, go back to the brain and see what are the configurations that are happening at those particular moments in time. This is usually called CAPS, co-activation patterns. And once you have the CAPS and their timing, similarly to what you would do with the functional connectivity states, you can start looking at how often they happen, which ones go from one to another, transition probabilities, and things like that. That makes sense? And this is just a few of the methods. There are even, and this is one good example to look at CAPS. Actually, this is the paper that originally described the CAPS, which also was done here at NIH, but by the group of Jeff Dunn. So let's look at a couple of examples just to go in more detail. Again, which method is better? It really depends what you're going after. If you think that it's a good assumption to think that the dynamics that are happening can be modeled as a succession of finite discrete number of states, then the idea of doing, a sliding window and applying some clustering, it makes sense. And here what I'm describing is what Elena Allen did, which is the original paper. So they did ICA, so they decided on getting their parcellation based on ICA, not a pre-existing atlas. They extracted representative time series for each of the networks. They computed sliding window correlation. They selected a window length, step, etc. And then they took all this connectivity matrix and applied k-mean clustering. They, in a data-driven way, decide how many different clusters they should look for, and they found these, I believe, 7 states. Because this was done on resting state data, it's hard to tell. I mean, it looks like there is a structure. It looks like it's different structure, but that's not enough to say this is real and this really being driven by something neuronal. So they were very limited. And then once you have this, you can do things like dwell times, look at how often they happen as time progresses. There are many different things that you can do once you have come up with these states. So the thing they went in a lot of different ways in interpreting the data. But for me, one thing that really stood out is this idea that they had this state that tend to happen more often the longer the scans were. And it's a state that the configuration that you get instead of connectivity tends to be the type of connectivity that you would see as people get drowsy. When you put that together with the idea that we know that people tend to get drowsy as scan progressive, it seems that they were finding something new, okay? But because we weren't sure about many other things, that's what we did the other study with the task to confirm that actually the different pattern of connectivity that we saw in the multitask, they really align with the different cognitive states that we impose via the task. Here is another study from that same group in which they look at the potential clinical value. In this case, I think they're comparing healthy control with schizophrenic, I believe, or bipolar. The idea being they apply this pipeline, they come with the states, they find them, and what they see is that some particular states are more common or more prominent in the healthy population or on the patient population. So it seems that looking at the dynamics from this point of view can be very clinically meaningful. This is a study that I like a lot. This is a study that they did in monkeys or macaques. in which they do a sliding window, functional connectivity analysis with a sliding window, but they have them with different level of anesthesia. And they find all different states. And what's really interesting is that the state that looks more like just the structural connectivity, more than these other ones, that's like something sitting on top, tend to appear much more prominently when the subjects are, or the animals are in deep anesthesia. So it looks like the dynamics that you see on awake are much more variable than the dynamics that you see when they get into a state of deep anesthesia, which again, I think it adds to the idea that what we see with this type of analysis, it's real. That said, there is a lot of detractors with this type of analysis. And there are studies that have shown that if you create surrogate data that has some of the properties of fMRI data, but they're stationary by definition, you can still get states that look like the one you get from resting state. And based on this observation and others, there's people who say that they think that what you see with dynamic functional connectivity is mostly a result of sampling variability, the idea that you, instead of having a single sample, you have many of them, they're noisy, so they're not gonna look the same. And that beyond that is basically just head motion and fluctuations in vigilance that's really driving that idea of dynamic connectivity. As you can imagine, I disagree with that, but I'm not the only one. There are also studies that have shown, in this case, they've been using in mice, they record both neuronal and hemodynamic signals all across the brain. This is not fMRI. And what they've shown is that when they do a sliding window correlation based on the hemodynamic signal or on the calcium signal, which is directly neuronal recording, they follow each other very well. This is for one particular connection. This is for another connection. Also, when you just not look at individual connections, but you do this business of a sliding window functional connectivity, there seems to be a strong agreement. And also, if you explore the data in terms of the caps that you get out of the calcium data and the hemodynamic data are quite similar. So you have both sides of the coin. It's still an open debate. I think nobody would disagree that the brain is not in a static configuration over time. The question is, what of that can you see with fMRI? And what do you have to do in your fMRI data to really capture the dynamics aspects that are related to that? That makes sense? So just a little bit more. As I said at the beginning, a lot of times it looks like functional connectivity, it's all about resting state, but it doesn't need to be. You can also look at functional connectivity with task data, whether it is naturalistic data or block designs. Now, when you're gonna do that, I think there are a couple of things that it's important to keep in mind. So let's go with the first one. So here is a study in which I think they scan 10 subjects, both during resting state and during a silent movie watching. We can take a sip on visual cortex, and this is a static connectivity, okay? And look at the correlation of the signal in that voxel to every other voxel. This is one representative subject, I believe, and we get, a connectivity map that makes sense. A lot of connectivity within occipital cortex, and then it looks like we have a little bit more connectivity with motor cortex. This is the same business done on a run that's the same length in which they were watching a movie. So when we look at this, it looks like the connectivity, although slightly different, but it's very similar. And that's something that we already know. And actually, I always like to get back to this quote. When we engage with a task, it's only small changes that happen over the spontaneous activity that is going on when you're not engaged. So to really get a task effect, it's hard from the point of view of connectivity, because the brain is continuously working, and it's just that that stimulus, that demand that we impose has a little change over it. That said, there is a change. And if we do the subtraction of the connectivity during the task minus the connectivity during resting state, what we actually see, I don't know if this is surprising for some of you or not, it's a decrease in connectivity overall from when they were from when they were watching the video to what they were resting. Now, this is one way to go after what's connectivity during tasks. I mean, two ways. I can just look at this, or I can look at this. Very different answer, no? Here is another one. So we can use this thing called intersubject, this is functional connectivity. What we're gonna do here We're just looking at the data from the subjects watching the video, and we're gonna pick a voxel in one subject, and we're gonna correlate the signal of that voxel in one subject to every other voxel in the other subjects. By doing that, what we're gonna find is the correlations that are due to the subject seeing the same video. No? The idea is that you'll have a spontaneous activity going on, but then because you're watching the video, there's gonna be some portion of the signal that is synchronized between you and the next person as they watch that video. So doing this kind of task, inter-subject functional connectivity, we're just gonna capture the connectivity that is really driven only by the task. And if we do that, we get a different map. Now, this is not true only when we look at a seed in visual cortex. If we do the same analysis just across the brain, very similar story. Those two are very similar. This seems to be a decrease in connectivity, and here we see a very constrained increase in connectivity in areas that are being driven by the movie, the visual cortex specifically. So how do we need to think about this? There's no right or wrong answer. It's how we interpret. So this one is clear. No? This one, I mean, resting state, whatever we see with the noise, our data perfectly is driven by a spontaneous activity. Here, it's going to be driven by a spontaneous activity and task-evoked activity. It's not that when you watch the movie, suddenly all other spontaneous activity stops. It's not like the brain, everything that it does is about the movie. It's doing a lot of other things. So when we're doing here, we're just getting connectivity that's based on both. When we do this business, under the assumption that your spontaneous activity and my spontaneous activity is not correlated, but the activity that is due to you and me watching the same video is, what we're getting is a connectivity matrix that is mostly driven by the task-evoked activity. And what we're getting here is something that has to do with both the task-evoked activity, but also the differential in spontaneous activity between when people were watching the movie and when people were not. And the negativity part that we see here, it seems to be associated, and those are very interesting papers, because it seems that when we engage with tasks, there is a quenching of the spontaneous component of activity. And that's why we see this decrease here. That makes sense? Now, the other thing to keep in mind when we think about functional connectivity from the point of view of tasks, and particularly we want to do functional connectivity based on event-related or block design, is this idea that because two regions suddenly go up in a block design above baseline, we're just watching a crosshair, right visual and left visual are like this, and then flickering checkerboard and they both go up. That doesn't necessarily mean that they're talking to each other. That means that the stimulus induces an activity on them. It might be that on top of that, they're talking to each other, but it's not an automatic one or the other. So let me make that point a little bit more clear, and this is an excellent paper looking at that. Let's say we have two regions, and their activity is correlated to the task time, the block design. But let's assume these two regions, they're not talking to each other. And we're gonna have several events, one block, another block, and then we're gonna look at the average time series and at the correlation of those. In a case like this, what we will have, is that different regions respond very similarly, although not the same, but you clearly every time have that kind of block-like thing. So when you average across all the occurrences, you get this, and definitely you see a correlation between these two regions that, as we know, they're not talking to each other. Why? Well, the way to think about it is because this is the result of two things, and the plus there, there are also nonlinear effects, like we just talked about with the quenching, but this is a simplification. You have this event-to-event response, that idea that the signal just goes up because the flickering checkerboard comes on. And then this other activity is happening in the background during the task. Now, in a case like this, in which they're not talking to each other, the only common signal is this, but this is not. As you can see from one event to the next, the signal is different in one ROI, but more importantly, it's different across both ROIs. If those were different, that's what would tell us that these two signals, on top of having that response, they're really going in synchrony beyond that. And if we were to remove this response, we would see that basically every time is kind of slightly different, because we can never really remove those responses 100%. There's still something, but the correlation comes down. If we go to another example where they're really talking to each other, which is represented here with this idea that even though across trials, the signal is different, the two regions within a trial, they're going with each other, in addition to that common response, then even after removing the task, we would still see that the correlation is there. So it's very important to find a way to remove those first level effects, that kind of, task-induced activity, just first level. If we really not want to make an inference only about likely active, but likely communicating. In this same paper, they go about a lot of analysis about how to do this properly. The take-home message here basically being, it's not possible to remove those just by using a canonical hemodynamic response. You're gonna need to come with a model or a way that takes into account that hemodynamic responses change across locations and across time. So something like a finite impulse response model or a constraint basis set seems to work pretty well, as they show here, because basically, this inflation that I just saw in terms of connectivity that is present when you do nothing, that you take part of it, but not fully when you use a canonical response. It seems to really go down when you use this either basis set or finite impulse response model, without really leading to a lot of false negatives. So that's all that I had for today. Just a summary, as I said, first thing, let's always remember that functional connectivity is just a statement about a mathematical operation that we do, and there are many reasons why that can be the case. So when you temperate your data, one can really be that these two regions are talking to each other, and very likely we do a lot of things properly, but always keep in mind that we're not really measuring that, we're just inferring that. We talk about different ways in which we can measure static connectivity, whether it's seed-based, ICA. One reason why potentially it's a good idea to look at it in terms of Pearson correlation and not something more complex, although there are other alternatives. The idea that for static connectivity, thinking about 10 minutes plus is a good idea, and to be very careful about things that can systematically induce patterns of correlation that might look appealing because they seem anatomically correct, but they're not driven by something neuronal of interest. We talk about the differences between dynamic and static connectivity, and this controversy about whether dynamic connectivity is really real or something that we should be spending efforts and time on. Many different ways to look at dynamic connectivity, and by all means, this is not, there are things like QPPs, complex PCA, you name it. There are a lot of other ways to look at this. And some considerations that you need to keep into account, extra considerations, you're gonna be looking at connectivity in task-based studies. So with that, thank you, and I'm open for questions. You want, Lucrecia, yeah, if you want to mute. Oh, no, sorry, that was just an applause. I thought it was a raise. Anything? Anything, anybody online? Well, I guess early lunch. You all know where to find me, or send me an email if you have any questions, or I can help you with your studies. Thank you.